Hello students, I am teacher Svetlana and in our today's lecture we will learn the biomolecules in a cell further in the enzymes. That is, today we will learn the mechanism of the enzyme action, factors affecting enzyme activity, and also learn the concept of metabolism, metabolic pool and secondary metabolites, which is covered in the chapter biomolecules. So let's begin. Mechanism of enzyme action. The basic mechanism by which enzyme catalyzes chemical reactions begins with the binding of the substrate or the substrates, that is more than one substrate, to the active site of an enzyme. Have a look at the image here. This here is the enzyme, and here we have the substrate approaching the enzyme. The active site is the specific region of the enzyme which combines with the substrate. So this here is the active site of the enzyme, and you will see that that site is occupied by the substrate, forming an enzyme substrate complex. The binding of the substrate to the enzyme causes changes in the distribution of the electrons in the chemical bonds of the substrate and ultimately causes the reactions that lead to the formation of the products. So here you can see that the products is formed and the enzyme product complex is now visible. The products are released from the enzyme surface to regenerate the enzyme for another reaction cycle. So here you can see that the products are released and the enzyme is free for another reaction cycle. There are two models to explain the mechanism of forming enzyme substrate complex. The first one is the lock and key model. Here, this is the image of a lock and key model. The specific action of an enzyme with a single substrate can be explained using lock and key analog, first postulated in 1894 by Emil Fischer. In this analog, the lock is the enzyme and the key is the substrate. Only the correctly sized key, that is a substrate, fits into the keyhole, that is the active site of the lock, that is the enzyme. So here you have the enzyme, this is the active site, this is the substrate. Only this shape or size of the substrate can fit in and form the enzyme substrate complex to give you the required products. So this is how it is compared with the lock and key, that only a specific key can open the lock, its own lock. Induced fit model, flexible model. Koshland in 1959 proposed the induced fit theory, which states that approach of a substrate induces a conformational change in the enzyme. It is more acceptable model to understand mode of action of the enzyme. Unlike the lock and key model, the induced fit model shows that the enzymes are rather flexible structures in which the active site continuously reshapes by its interaction with the substrate until the time the substrate is completely bound to it. It is also the point at which the final form and the shape of the enzyme is determined. So in this induced fit, we are trying to understand that the substrate has the active site which is flexible. You can see that initially the shape is like this, and then it allows the substrate to fit in and it reshapes itself, forming the enzyme substrate complex, and the rest of the activity remains the same. The complex binds and the products are formed and released. So the induced fit model supports that the enzyme is quite flexible, unlike in the lock and key model where it supports that the enzyme is a very rigid structure and has a very rigid active site which only allows a specific substrate to bind to it. Now students, let us have a look at the factors that affect enzyme activity. The first is the concentration of the substrate. Increase in the substrate concentration gradually increases the velocity of enzyme activity within the limited range of the substrate level. So what this means is that when the concentration of the substrate increases, the enzyme activity gradually increases, but it remains within a limited range of the substrate levels. A rectangular hyperbole is obtained when the velocity is plotted against substrate concentration. Have a look at this graph. This is the increase in the substrate concentration and this is the increase in the rate of the enzyme activity. You will see that as the substrate increases, there is a gradual increase in your enzyme activity also, but it is in a limited range. Beyond a point, there is no increase in the enzyme activity seen. Three distinct phases A, B, and C of the reactions are observed in the graph. So these are your A, B, and C region. First, there is steady and constant growth that is happening till it reaches its maximum velocity. That is the maximum rate of reaction that can be reached. And then your C is the static phase, where it from here on there is no further increase in the rate of reaction even if the substrate concentration increases. Here on this graph you can see V, which is indicates the measurement of velocity. V max is the maximum velocity, and S is your substrate concentrations, and Km that you can see here is called as Michaelis-Menten constant. Km, or the Michaelis-Menten constant, is defined as the substrate concentration expressed in moles per liter to produce half the maximum velocity in an enzyme catalyzed reaction. So Km means the amount of substrate that is required to produce half the maximum velocity. That is, this is the maximum velocity, half the maximum velocity to produce the amount of substrate that is required. That value is called as Km value. Now, if it indicates that half of the enzyme molecule, that is 50%, are bound with the substrate molecules when the substrate concentration equals to the Km value. Km value is a constant and a characteristic feature of a given enzyme. It is representative for measuring the strength of the enzyme substrate complex. A low Km value indicates a strong affinity between enzyme and substrates, whereas high Km values reflect a weak affinity between them. For majority of the enzyme, the Km values are in the range of 10 to the power minus 5 to 10 to the power minus 2 moles. The next factor is the enzyme concentration. The rate of an enzymatic reaction is directly proportional to the concentration of the substrate. The rate of reaction is also directly proportional to the square root of the concentration of the enzyme. It means that the rate of reaction also increases with the increasing concentration of the enzyme, and the rate of reaction can also decrease by decreasing the concentration of the enzyme. So here you can see it is a simple graph. So if you have your concentration of enzyme increasing, obviously the rate of reaction is going to increase, and if the concentration of enzyme is decreasing, then the concentration with the decrease in the concentration of the enzyme, the rate of the reaction is also going to decrease. Then we have temperatures. The enzymatic reaction occurs best at around 37 degrees Celsius, which is the average normal body temperature in homeotherms. The rate chemical reaction is increased by a rise in temperature, but this is true only over a limited range of temperature. Enzymes rapidly denature at a temperature above 40 degrees Celsius. The activity of enzyme is reduced at lower temperatures. The temperature at which the enzyme shows maximum activity is called as the optimum temperature. So you can see, with increase in temperature, you have an increase in the enzyme activity till it reaches its optimum temperature, which is around 40 degrees Celsius. And beyond that point, you have a temperature. If it increases further, then the enzyme activity falls down drastically. Effects of pH. Similar to the temperature, there is also pH at which an enzyme will catalyze the reaction at the maximum rate. Every enzyme has different optimum pH value. The enzyme cannot perform its function beyond the range of its pH value. So the graph is similar to your temperature graph. There is an increase in the rate of reaction with the increase in the pH value only till it reaches its optimum pH. Once the pH increases beyond that, then the activity falls down. Then we have other substances. The enzyme action is also increased or decreased in presence of some other substances such as coenzymes, activators, and inhibitors. Most of the enzymes are combinations of coenzymes and an apoenzyme. Activators are the inorganic substances which increase the enzyme activity, and on the other hand, inhibitor is a substance which reduces the enzyme activity. Now students, let us have a look at the concept of metabolism. Metabolism is the sum of chemical reactions that take place within each cell of a living organism and provides energy for vital processes and for synthesizing new organic materials. It involves continuous process of breakdown and synthesis of biomolecules through chemical reactions. Have a look at this image here. Here you can see it is your anabolic reaction, where you have smaller molecules and energy coming together to give you a larger molecule, and here you have the catabolic reaction, where you have the larger molecule which is broken down into smaller molecules and energy is given out. So either there is a breaking down of the molecule, or there is synthesis, that is formation of a new molecule in your metabolism. So we have catabolic pathways. It leads to the formation of simpler structures from a complex biomolecules. Example, when we eat wheat, bread or chapati, our gastrointestinal tract digests, hydrolyzes the starch to glucose units with the help of enzymes and release energy in the form of ATP, that is adenosine triphosphate. Anabolic pathway is called as biosynthetic pathway that involves formation of more complex biomolecules from a simpler structure. Example, synthesis of glycogen from glucose, proteins from amino acids. These pathways consume energy. Then we have something called as metabolic pool. Before that, you can have a look at this image here. This shows the catabolic reactions and this shows the anabolic reactions. Here you can see complex substances are broken down, and here you can see from simple substances the complex substances are formed. In the metabolic pool is the reservoir of biomolecules in the cell on which the enzyme can act to produce useful products as per the need of the cell. The concept of metabolic pool is significant in cell biology because it allows one type of molecule to change into another type. Example, carbohydrates can be converted to fats and vice versa. The metabolites can be added or withdrawn from this pool according to the need of the cell. The balance between catabolism and anabolism maintains homeostasis in the cell as well as the whole body. So it maintains our overall well-being collectively, both the catabolic and the anabolic reaction. So metabolism is a very important phenomenon that takes place in every living cell. Then we have secondary metabolites, SMS. Secondary metabolites are small organic molecules produced by organisms that are not essential for their growth, development and reproduction. Have a look at this image here. You can see the primary metabolites include carbohydrates, proteins, lipids, nucleic acids and hormones, and the secondary metabolites are the ones that we are going to discuss ahead, which are not essential for the growth and development. Secondary metabolites can be classified on the basis of their chemical structures, that is, they contain rings, sugars, composition with or without nitrogen, their solubility in various solvents, or the pathways by which they are synthesized. A simple way of classifying secondary metabolites include three main groups. Terpenes, made of mevalonic acid that is composed mainly of carbon and hydrogen. Phenolics, made up of simple sugars containing benzene rings, hydrogen and oxygen. Nitrogen containing compounds, extremely diverse class, may also contain sulfur. I have listed here the different types of secondary metabolites and their roles. You can pause the video and have a look at it. Economic importance of secondary metabolites. They are used in the treatment of infectious diseases, cancer, hypertension, inflammation. Morphine is used in pain relief and a cough suppressant. Then we have alkaloids like nicotine, cocaine and terpenes like cannabinoids, which are used for recreation and stimulation. Flavors of the secondary metabolites improve our food preferences. Then we have characteristic flavor and aroma for the cabbage and its relatives are caused by nitrogen and sulfur containing chemicals, plus it also gives protection for the plants from many pests. Tannins are added to wines and chocolate for improving astringency, and some of them also have antibiotic properties, and some of them are used as food preservatives. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.